Welcome everyone to our webinar. This webinar is a collaboration between Molecular Devices and Greiner Bio1. We will be talking today about the emerging induced pluripotent stem cell applications in drug discovery. My name is Kasia Proctor and I will be your host today. Let me introduce our speakers. Rainer Heller will be our speaker. He is a product manager from Greiner Bio1. He's been with Greiner for over 12 years now. Following Rainer, we'll have Kathleen Salamo. She is our application scientist in Europe. She has joined Molecular Devices in 2011. And then Christian Holtz will be our final presenter, and he's an application scientist also with Molecular Devices, and he's been with the company for over 5.5 years. With that, I will hand it off to Rainer.
So thanks, Kasia, for the kind introduction, and hello everybody and welcome to our today's webinar. As introduction, we thought to start with honoring today the Nobel Prize winners in medicine or physiology of this year. This Nobel Prize was given to John Gurdon and Shinya Yamanaka for the discovery that major cells can be reprogrammed to become pluripotent. Especially fascinating about this finding is that now pluripotent stem cells, so-called induced pluripotent stem cells, can be generated from human somatic cells, including disease-derived human cells. And major cells, including nerve, heart, liver cells, can be derived from induced pluripotent stem cells, allowing scientists to study disease mechanisms in new ways. As the use of human-derived real embryonic stem cells in research is quite restricted, induced pluripotent stem cells, they really do offer an alternative, especially for drug discovery research. Human-induced pluripotent stem cells are meanwhile commercially available, and our attempt was to test such cell lines in practical approaches. And today, we would like to show you our experience and results in this webinar. This agenda gives an overview about the different sections of the webinar. We will start with a very short introduction, a short technical summary about the scientific basics of induced pluripotent stem cells, and an explanation why we selected neuronal cells as model. Then we will describe our experimental approaches, where our experience in maintaining, growing, and differentiation of two human iPS cell lines is summarized. In the next section, the used materials with a focus on devices and microplates are described. The obtained results will then be discussed by first describing each experimental approach, followed by the data analysis, which was done either in a high throughput manner, a high content screening manner, or both. And with a short summary and outlook, the webinar will end. So the next slide. Induced pluripotent stem cells were produced first in 2006 from mice and in 2010 from human cells by the team of Shinya Yamanaka. Somatic cells, for example fibroblasts, were forced to express the embryogenic relevant genes of 3-4, SOX2, KLF4, and thymic by a viral infection. These reprogramming genes, which are considered as oncogenes, lead to dedifferentiation of somatic cells into embryonic-like cells then. iPS cells are believed to be similar to natural pluripotent stem cells, such as embryonic stem cells, but the full extent of their relation to natural pluripotent stem cells is still under investigation. Meanwhile, rapid progress has been made in the last years and nowadays. Nevertheless, it must be said that the therapeutic use of induced pluripotent stem cells is still far off. However, for us in drug discovery, induced pluripotent stem cells, they really offer a great opportunity as they can be human, not only animal-derived. And due to their great plasticity, they can be differentiated in virtually all cell types of an organism, and there are no ethical or legal restrictions, unlike the original human embryonic stem cells. Furthermore, an almost unlimited pool of source cells for various specific human iPS, including disease-specific iPS, for new experimental approaches is available. And last but not least, induced human embryonic stem cells are relatively easy to maintain, to handle, and to propagate. But why did we select neural cells, or better to say, human IPS differentiated into neurons as a model. The main reason was the interest of many scientists in this kind of research, which is used to study neurodegenerative diseases. In an aging society, such diseases like Alzheimer, dementia, or Parkinson do play an increasing importance, and an efficient treatment would be doubtless a major progress in medicine. The two pictures on the slide show an example of a neural outgrowth assay, which is a typical test used to screen compounds mediating activation of microglial cells. And in the later slides, you will see that we have tried to do something similar. The images here on the slide were kindly provided by the Max Planck Institute for Molecular Biomedicine in Münster. The used disease model is amyotrophic lateral sclerosis, ALS. In ALS, the aberrant activation of microglial cells plays an integral role in the chronic phase. Therefore, agents mediating toxicity of microglial cells may delay disease progression. So the left image shows neurons growing on non-inhibited microglial cells. The suppressed development of the neural cells is clearly demonstrated. So you see here on the left side the cells where they are not feeling really healthy. The right image shows neuronal cells growing microglial cells with suppressed activation. The neurons form dendrites and do show a higher vitality. A disease model, mouse embryonic stem cells were grown on black microglia 284 microplates, and the analysis was done including the detection of branch points, the length detection or the measurement of the length of axons, and the survival of the neurons. The next slide gives you an overview about our experimental approach. The first experimental approach was the use of a human foreskin-derived iPS cell line, which we purchased from System Biosciences. We tried to differentiate this iPS cell line into neurons for further testing. The second experimental approach was the use of a pre-differentiated cell line from cellular dynamics. These so-called I-cell neurons are a mixture of post-mitotic neural subtypes comprised primarily of GABAergic and glutamergic neurons with typical physiological characteristics and responses. In parallel, or as a control, the live laser, a beta-lactamase reporter gene assay from Invitrogen was conducted. In this assay, the stimulation of a G protein-coupled receptor is detected. The test is based on CHOK1 cells expressing the human acetylcholine subtype 1 receptor, the M1 muscarinic receptor. So, the cultivation of IPS, of human IPS. This slide shows the workflow for generating neurons from a human IPS cell line, which we obtained from System Bioscience. I do not want to go into details about the different workflow steps, as this would take much too long, but I would like to name some of the some of our obstacles we faced. First, the whole process was extremely time-consuming. It took us nearly 2 months until we received the first differentiated cells and results. Furthermore, the generation of neurons from scratch was very labor-intensive and expensive due to the need for daily time-consuming media change over 2 months. After our experience, we classed the first IPS approach as non-idle for routine drug discovery tests. And we switched to the I cells. And the I cells, in contrast, these are pre-differentiated neurons from cellular dynamics, and this cell line was really quite convenient to use. The results were obtained after 10 days, as no pre-culture is required, and there is no need for an expensive special media, such as the matrix gel, which was used for the foreskin-induced pluripotent stem cells, and the cells were seeded just directly on the PLO laminin-coated microplate. The media change is only necessary every 3rd to 4th day, and therefore the I cells were our preferred model for the subsequent testing. So let's move to the section about used microplates and substrates. For all tests, black clear film bottom microplates were used. The neurons require polyornithine coating with a subsequent laminin coating. We tested also cell culture treated or poly-D-lysine coated microplates, but we found the best performance with the originally recommended polyornithine laminin coating. And this slide here gives an overview about the different available surfaces with microplates. First of all, there are center plates or center plate plus. These are glass bottom microplates. These glass bottom microplates consist out of polystyrene frames with a glued 175 micrometer thick glass bottom. Glass is an amorphous, non-crystalline solid material which is brittle and optically highly transparent. The refractive index of standard borosilicate glass is typically 1.523. Glass is the gold standard in high-resolution microscopy. However, coating of glass is very, very difficult, at least for us, and we would not recommend glass-bottom microplates for the sensitive IPS cell lines. Second, there is a large range of microclear plates, polystyrene microplates. Microplate is our microclear. This is our Greiner brand name for black or white clear bottom microplates, which are manufactured completely out of polystyrene. Polystyrene is a semi-crystalline to amorphous material. It is highly transparent with a limited flexibility and a refractive index of 1.51. The microclear microplates, they have typically a 190 micrometer film bottom and are with different cell culture treatments available. Cell culture treated microplates would be our recommendation as well for the polyomethylene laminin coating, which is required for the IPS-derived neurons. And finally, there is a new product line which is called ScreenStar. The ScreenStar microplates are manufactured out of cycloolefin with 190 micrometer cycloolefin bottom, and these cycloolefins are amorphous polymers with glass-like characteristics. So cycloolefins do have excellent optical properties with low light absorbance rates and low autofluorescence in the UV. Their refractive index is with 1.533, very close to the refractive index of glass. And the plates are cell culture treated available, which is again the best performance in the required polyomethylene laminin coating process. Our focus for the assays was on 1536 cycloolefin microplate in order to demonstrate the performance of this new microplate surface in a high throughput screening approach with a sensitive cell culture system. However, all tests were done in parallel in classic polystyrene clear film bottom plates. And the next slide, I need the support from Christian from Molecular Devices. Christian, can you help me?
Yeah, so here you actually see three images from three different kind of plates. On the left-hand side, the Greiner ScreenStar plates with the cycloolefin surface. In the middle, you see the microclear plates, which the polystyrol thin bottom, and also on the right-hand side, we compare that to a competitor plate. Now, the images here are all acquired with the Image Express Micro in the Derby blue channel with a 60x objective. It doesn't show any cells here. We just wanted to make clear here that the autofluorescence, which actually comes from these plates, is actually really low in the Greiner ScreenStar plate. So in comparison to the polystyrol plate and competitor plates, you can see the average intensity when we just focus into the wells here, they're much reduced.
Okay, thank you, Christian, for the support. And yeah, because of the excellent performance of the cycloolefin material in microscopic applications, Greiner decided to enlarge the product range by the ScreenStar of the ScreenStar brand, which includes microplates in 96 well, 284 well, and 1536 well format. The 1536 well ScreenStar microplate is already available. And the 96 well plate we will launch at the SLS beginning of next year, and the 384 well ScreenStar microplates are in beta test. So if you would like to receive some samples, let us know. Common for all ScreenStar microplates is their recessed film bottom. And due to the small gap between the plate bottom and the microplate rim, very low working distances in microscopy are possible. Therefore, high magnification objectives as well as oil or water immersion objectives do not need to be moved in the z-axis over large distances. And objectives with different magnification levels can be changed during the microscopic analysis without touching the microscope table. And I would like to come to the end of this first section of the webinar with this last slide, showing the performance of 96 well ScreenStar microplates in a non-confocal microscopic application. For the test, we stain simply cells, or the first grown cells, on 96 well ScreenStar plate surfaces and stain it afterwards with DAPI and phalloidine. Then the imaging was done with oil immersion and a 63-fold magnification with a Leica fluorescence microscope. And I think you can really see it, and it's obvious that the pictures made with screen star microplates show much higher resolution compared to pictures made with classic polystyrene film bottom plates. So now it's Kathleen's turn, and she will say more about the devices used for the analysis, and then we come at the end to the data and data description. So Kathleen, please go ahead.
Thank you, Rainer. So in the next section, we would like to talk about the instruments as well as the results. So we would first like to present the benchtop instruments which were used from molecular devices for the experiments that we have performed. So Christian will later explain the Image Express Micro XL, which is an automated microscope from molecular devices used in high content screening. And I would like to give you an overview of the Spectramax paradigm, which is an upgradable plate reader from molecular devices. So to start with, the Spectramax paradigm is an upgradable plate reader, so you can add new applications to it at any time in the future whenever you need a new application. With the two integrated photomultipliers, you can easily perform dual wavelength experiments in a very fast manner and also increasing your data quality at the same time. You can read from the top and from the bottom, and you can increase your sensitivity of your assay with adjusting the focus point of the light beam in the set height. As well, you can read from 96 well plates to 1536 well plates for high throughput experiments. And on top, we do offer a mode where you can do fast reading, which is called on the fly, and which I will explain later in my presentation. And to complete this, the paradigm is ready for automation solutions. It can easily be integrated with a plate stacker like the Spectramax from molecular devices or with any other liquid handling system. So, as I already said, the paradigm is a flexible system, and this is achieved by its unique modular design. So, on the one hand side, you have a base unit, which includes the detection system, the plate handler itself, and environmental control. And on the other side, you have cartridges to customize your reader. These are optical cartridges. They do contain light sources, monochromators, and filters. And we do have both multifunctional cartridges as well as assay-optimized cartridges available. And then those cartridges are added into the top or the bottom position of the reader. This can be easily done within 2 minutes. So you have 6 positions in the top and 6 positions in the bottom. And in total, this allows you the most flexibility in your lab for your assays. And you can add any newly developed cartridges for molecular devices at any time in the future because the base unit itself does not require any changes for that. If we now take a look into the cartridge itself, so what I did here is actually taking a gene placer cartridge for dual wavelength detection and sliced that through in the middle. It's shown here as a cartoon, and you can see that we have a high power LED in there, and from there the signal is transferred to a first filter, and then the one part of the light is going to a photodiode for signal normalization. And as well, the intensity of this high power LED can easily be triggered and adjusted to your sample concentration. So this excited light goes then onto a dichroic mirror. Then you see the red arrow, so this light is then going into your well. The sample gets excited, and then the emitted light is going back into the cartridge. Then, since we have a dual wavelength cartridge here, the first wavelength is then reflected onto your first PMT, and the second wavelength is then reflected onto your second PMT. So it's important to say that we do have the option of dual experiments inside the reader, which you can read at the same time, not one after another, which is quite convenient for thread applications, fluorescence, polarization assays, as well as other dual emission assays available. Coming to the dynamic range for in fluorescence assays, I'm showing here an example of a fluorescein dilution series, which I have performed, and we can easily achieve with fluorescein a 7-log dynamic range, and that's one advantage of the systems, and this is performed because you have these high power LEDs in there, and we do trigger the sensitivity of the high power LED rather than adjusting a PMT, which is very slow, and matching those a fixed PMT gain together with the adjustment of the LEDs and LED intensity brings us to 7-log dynamic range. In the next 2 slides, I would quickly want to go over the Softmax Pro software, which is our data, so it's our reader control software as well as an analysis package for your data. So it's GXP ready. So if you are working in a GXP environment or you want to transfer your assays to a GXP environment, you can easily use that software. So you do set up your settings, and then your data is being collected in the plate section, which you can see here. So the numbers are displayed. And this data can also be viewed in a way that you have your numbers, of course, but also a color gradient or as a 3D map as it's shown in this image here. And now you could export your data in Excel or a text format, but you could also go for a next step and do data analysis and statistics on the software. So the first thing is you do consolidate your data into tables grouped by their sample type, like standards, controls, unknown samples, and then you can calculate the mean, median, and CV values, as well as customizing these tables by using other statistical formulas or queries to do quality assessment of certain parameters. Then this data can be transferred to a graph section. You can apply multiple curve settings there. So we do have several options like 4P, 5P curves, linear curves, bi-exponential curves. So it's a wide variety in there. Then underneath the graph, you can see also the curve fitting parameters as well as statistical parameters. And those can be later transferred into a report. So you can add in your experimental section a report type where you can add in your data, your SOP, but on top you can generate a summary report with all parallel line analysis results and show the potency of the different curves just as a short summary part in your report. And all these protocol sections can be saved as a template, and you can reuse them for future measurements and just import new data into these setup reports. And with this, I would like to hand over to Christian.
Thank you. I would like to show you just a couple of slides for the Image Express Micro. So the Image Express Micro is an automated high-content screening instrument. It's a benchtop microscope, and it uses a laser autofocus for really fast focusing on your sample, on your plate. You can also add like an image-based focus towards it. Now, this instrument is plate format independent, which means like you can actually use any kind of plate type, so from any kind of manufacturer, but also any kind of format, from a 1-well plate up to a 1536-well plate. You can equip this instrument with anything from a 1x objective to a 100x objective for various kind of assays. And also what's new in that instrument now is that we have a really large camera chip, which I'm going to show you in a moment. And we also have equipped it with an LED light source. And we have a new software drop-in, which is like digital confocal, which is there for a really fast deconvolution algorithm during the acquisition. Now, looking at the large field of view, this is an image from our camera, from our standard camera chip, and then you can compare that to an image from a large camera chip. You can see that it's about 3.5 times as big. So this actually has the advantage that you cover more area. You're also much faster in the acquisition, and you have less objects on the edge of the image, which means like there's less risk of cutting images or objects in pieces. So in this sense, it's ideal actually for acquiring neural networks. Down here you actually see the camera, and also down here you see the 384 well. And if you would acquire that with 4 objectives, you actually cover the whole well with that magnification. Now, we also have equipped this microscope now with an LED light source. So the advantage here that you have a much longer lifetime, so over 15,000 hours. So the LED light source is as bright as a ceiling lamp. You don't need a shutter. So you have here the electronical shutter, and this light source has a high stability and also no degradation over time. And it still covers the whole range from the blue to the red wavelength. Now, our software drop-in here, Digital Confocal, is there to really clean up the images on the fly during the acquisition. So on the left-hand side, you see here an image of epithelial cells without Digital Confocal. On the right-hand side, you see the same image with Digital Confocal. And especially here in the red with the tubuline, you see this gives much better results, which is then much easier to actually analyze with the image analysis software. Now, what's also new is that we have a very neat and nice custom module editor. So this is for image analysis. So this is a drop-in function that you have an automated workflow where you can simply say which kind of objects you want to identify, for example, nuclei, and you want to identify granular structures around the nuclei or within the nucleus, and it's very easy to use. So this workflow, you always see like, which steps you're actually doing. So you create this kind of custom module, and then you can actually run it in our image analysis software, MapExpress, for all your plates, for all your samples. Now, the ImageExpress Micro has different kind of hardware options. So on the left-hand side here, you see environmental control. So the whole upper part of that instrument is temperature controlled, but also CO2 controlled, and also it has humidity control. Not only keeping the sample at a certain temperature, but also the optics, so you don't have any kind of pixel shift. Now, we also offer a transmit light option. So this is like a real condenser, so you can color your light, and it also offers phase contrast. So in this sense, you can also acquire some cells which are actually not labeled. So label-free technology here. On the right-hand side, you see the ImageExpress Micro being equipped with fluidics. So this is an online pipetter, which is often used for calcium influx assays. So the ImageExpress Micro is actually in a whole complete solution that we do offer. So this is driven by software called MapExpress. And MapExpress saves the images in a database, so in a very organized fashion, from where you can retrieve them, again, the images, and do some image analysis with our application module or the custom module builder. The image analysis results are saved into the database again, from where they can retrieve again with our Acuity Express software, which is a cellular informatics software. So here you can do your statistics and your graphs. Now we have to use the software for acquiring some images for the Mito ID assay, but mainly this assay has been now processed and acquired with the Paradigm instrument. And Rainer will say a few more words about the Mito ID assay.
Yeah, thank you, Christian. So the Mito ID, this was the first kit we used to detect the growth and the behavior of the IPS, the neurons, IPS-derived neurons, on the surfaces, yeah, we use both methods, so the paradigm and the image express, but the focus was on the paradigm. And the MitoID membrane potential cytotoxicity kit measures the fluctuation in mitochondrial membrane potential utilizing a cationic dual emission dye that exists as green fluorescent monomers in the cytosol and accumulates as orange fluorescent J aggregates in the mitochondria. In the mitochondria, having a low membrane potential will accumulate low concentrations of dye and will exhibit green fluorescence, while more highly polarized mitochondria will exhibit orange-red fluorescence. And thus, exhibit a shift from orange to green fluorescence as mitochondrial function becomes increasingly compromised. The advantage or the special thing about the MitoID is a live cell dye, so it does not need any fixation steps, and later on I will say something about fixation of cells. And furthermore, the signal is very, very strong, so that you can use the MitoID in microscopy, but as well in a fluorescence reader. Okay, Kathleen will continue with the presentation and show you some results.
Thank you, Rainer. So, in this slide, I'm showing the results of the 1536-well screen star plate that we have measured on the paradigm using our Tune cartridge, which is a flexible fluorescence reader cartridge. So we have plated the cells in a way and treated the cells with staurosporine increasing to the right, as you can see in the small cartoon, showing you the signal intensities from the orange signal only. I'm only plotting the orange signal from the 590 nanometers. And as expected, that goes down with increasing staurosporine concentration because it also destroys the membrane potential. And the plot below shows you a 4-parameter log curve. So again, the orange signal plotted against the concentration of the staurosporine, and we could nicely determine an IC50 of 0.042. And coming to the next essay, Rainer will shortly explain you the live laser.
Yeah, the live laser was used as a kind of control. The live laser is a FRET assay based on a gene placer CHO cells, and they contain the acetylcholine receptor, and this GPCR is daily integrated into the so-called cell sensor beta-lactamase reporter gene construct. Everything is available from in vitro gen. Without GPCR activation, very simple, the beta-lactamase remains inactive, and the fluorescent substrate emits the green fluorescent signal at 520 nanometers. After GPCR stimulation, the beta-lactamase becomes activated and cleaves the fluorescent substrate into dyes and disrupts thread, so that exciting at 409 nanometer produces a fluorescent signal at 447 nanometer. And this fluor signal can be readily observed in a microscope or in the paradigm as a microplate reader. So Kathleen will show you the results we received with the beta-lactamase reporter gene assay.
Thank you, Rainer. So I would like to show you the results of the live laser on the paradigm using a dedicated cartridge for the live laser. So we do have a cartridge called the Gene Laser, which has exactly the filter sets in there which you need for this assay and is optimized for this kind of assay. So in the upper cartoon, you see the plate layout. We have plated the stimulated cells on the left-hand side with an increasing copper hole concentration to the right, as well as unstimulated cells in blue on the right-hand side and background cells in red. Once the plate reader has read the data, those data is populated in the data section of the plate, and you will then see two values. The first one coming from the first filter set with an emission filter at 465, and the second one has an emission filter at 535. And this data was then transformed into a ratio as described in the literature, what you use for the live laser assay to view your results. And As a first experiment, we wanted to look at the plates, at the differences of the plates, how those experiments would look like. So you do see different plots here. Again, the ratio against the concentration plotted with a 4-parameter fit. And finally, we did compare the EC50s, and I also documented the Z values. And for the plates that we have tested, screen style, the PS plate, and the competition plate, which Rainer initially introduced, we could not see any significant differences, which we had already expected here. And as a next experiment, we took one of those plates. We used the PS microclear plate 1536 well, and had the goal to compare different read modes on the reader. So we do have a stop read mode, as well as the on-the-fly mode, which I introduced at the beginning, where you can increase your speed. So what you see is that with a stop read, you will read your plate at 6 minutes, but if you switch to on-the-fly, you can achieve a read time of 1.5 to 1 minute. So what does it mean, stop read? If we look at the cartoon for the read pattern, what actually happens, the measurement of the signal is done. The measurement of the signal is done when the well center is positioned right under the read head and it's not moving anymore. So when it's stopping, and then the signal only is detected. And with the integration time, the user has actually said, in this experiment, I have used 140 milliseconds. If I then switch to the on-the-fly mode, the read pattern changed. So what actually happens is that while the plate carrier is moving, the signal will be detected, as you can see it in the cartoon below. So we do differentiate between two on-the-fly modes, as I specify here. And one is optimized for speed and is designed to obtain the fastest possible read time, which is 1 minute for a total 1536 well plate. And on the other hand, we do have it optimized for performance, which reduces the read speed slightly to the speed on the fly mode. And we then end up with a read of 1.5 minutes for a total 1536 well plate. And now comparing the EC50 values from this experiment for the different read modes, we actually found very similar results, and this would suggest us that we can use the fastest possible read time for this application and increase the speed to run this total 1536 well plate. And with this, I'm handing over to Christian again to explain the results from the microscope.
So we have used the same kind of assay, the same kind of plate to acquire with the Image Express Micro. So here we used the 40x objective to get a little bit more resolution. And we also used the Spectramax plate, so the screen style plate, to have like a really nice signal to noise. So here you see actually two montages of images from different kind of wells. So in the upper image in blue, you actually see coumarine in the blue channel, and in the lower montage you see fluorescein. So that is in the green channel. And here you see, like, from the right going to the left, you see the couple coil stimulation. Now, these images can be also montaged, and click on that. These images can be also montaged together. Now, if I now zoom in into the images on the right-hand side, you see actually quite a lot of these blue cells, whereas on the right-hand side, where it's unstimulated, you still have like a higher concentration of just like, blue of green labeled cells. Now, these images can now be analyzed with our image analysis software. So looking at this image again, and looking at the two different kind of wavelengths, we actually used one of our image analysis application module called multi-wavelength cell scoring. So this is in the MetaXpress software. And what you do here is you just select the number of wavelengths. So in this case, we have only 2 wavelengths here. And we can then define, like, the intensities and the sizes of our objects, and where we actually set certain kind of intensity cutoff. Now, if this analysis is run over our images, you can see how many cells are actually counted in one wavelength and in the other wavelength. So in the end, we actually get a cell-by-cell table. For every single cell, if you click on this one, you actually see it in a cell-by-cell table. What is actually the intensity in the blue channel or in the green channel, the average intensity of that object. Now, if you compare that over the whole plate at different kind of positions, we could calculate the ratio between the aquamarine and the fluorescein, and you see, like with the simulation here on the left-hand side, we have a much higher ratio as if we have here on the right-hand side with the unsimulated cells. Other readouts we can also look at are, for example, the average intensity of our cells in the blue channel, or also the percentage of positive cells. So basically, you set like a threshold and you count how many cells will be over this certain kind of threshold in the blue channel. Now, what you then can also do is you can annotate your plate in our Acuity Express software. So you can say on which position you have which concentration. So here on the left-hand side, the highest particle concentration. On the right-hand side, you see the lower concentration, or just the control with the DMSO. And we can then load that into our software, into your Acuity Express software, and calculate the EC50 after doing curve fitting. And the EC50 was very similar as we had with the Paradigm instrument. So that also shows that those instruments